In evolutionary biology, there are so many concepts, one of which is life history. So you might ask, what is life history? Life history describes the patterns of survival span, growth, and reproduction of an organism. Life history traits include development rate, reproductive scheduling, parental investment, body size, and number of offspring produced. These traits influences the reproduction and survival chances of a species. So what are the factors that can influence life histories? One of these factors is hormones expressed in different species. Studies have been conducted on hormones such as insulin-like growth factor 1, or known as IGF-1, which is an endocrine growth hormone produced primarily by the liver. Increase in IGF-1 is correlated to accelerated life histories across 41 mammalian species. This is characterized by increase in reproductive and growth rates, but decrease in lifespan. Based on their research, my research focuses on estrogen, a female sex hormone that affects the development of reproductive organs and induces self-proliferation and differentiation. This project serves to study the relationship between the levels of estrogen expressed and variations in life history traits across mammalian species. Not many studies have been conducted to study hormonal pleiotropy and how it affects the variation in life history traits across species in females. So why is it important to study estrogen? The relationship can be used to predict life history traits of new mammalian species. The hypothesis of this project is that heightened levels of estrogen are associated with faster life history patterns in female organisms. This means that these species have faster reproduction and growth rates but shorter lifespan. 22 female mammalian species will be selected for the study. Based on previous database and literature reviews, the average levels of estrogen at the peak of maturity for each species are recorded together with the life history traits. The traits that are analyzed are age at sexual maturity, number of offspring produced, maximum lifespan, average gestation length, and average body mass. SPSS software is then used to plot linear regression models where the level of estrogen is plotted against individual traits. Each point on the graph represents a species at the peak of maturity. The closeness of the relationship between the two variables can be determined by using the Pearson product moment correlation coefficient. The sign of this coefficient represents the positive or negative correlation, while the magnitude represents the closeness of the two variables. So what are the benefits of this research? Based on this research, the scientific community can conduct further research on how different hormones and other factors interplay to influence life histories to emphasize complexities behind this relationship. Predictions of life histories can be made for new species that will be discovered in the near future. This study can also highlight the differences in life history patterns which can be made known to the general public as they may not be aware on how and why these patterns differ across species. The public can also learn more about how different factors play a part in influencing such patterns and provide a clearer picture on why different species have different phenotypes. Hopefully this study invokes interest in evolutionary biology.